welcome to the program. We thank you very much for joining us. I'm Anna Meyer. I'm a cardiac surgeon at Heidelberg University, Germany, and I'm the leader of the MCS program. We're excited to bring you today's program, Xenotransplantation, Crossing the Final Frontiers. My co-moderator today is Simon Urschel. He is a pediatric cardiologist at University of Alberta and member of the Interdisciplinary Network for Advanced Heart Failure and Heart Transplantation. We would like to acknowledge and thank our partner level supporter, Theracos Institute. This webinar is made possible through their generous support. The recently performed heart transplantation using a genetically modified pig heart into an adult recipient has catapulted the discussion around xenotransplantation back on the center stage. After decades of research, grown understanding and ability to overcome interspecies differences in animal models, have we now reached the point of justifying xenotransplantation into humans? The program includes expert presenters who will provide their perspectives to this question. During the presentation, you are encouraged to submit questions. After the presentation, there will be a panel discussion that will include all the speakers as well as additional experts. It is my pleasure now to introduce our first speaker, Dr. Richard Pearson. He's also a cardiac surgeon and a scientific director, Center for Transplantation Science, Massachusetts General Hospital in Boston, USA. Dr. Pearson, welcome to the program.
Thank you. It's a pleasure to be here today. The charge I've been given today is to share with you the genetic engineering approaches that have been taken to overcome the barriers to transplanting pig organs into humans, as has recently been demonstrated at the University of Maryland. Those of us who've been working in the field are well familiar with these barriers, but perhaps for this audience, it is worthwhile to take a moment to step back and think about why we're even pursuing this approach. The advantage of the xenotransplantation, should we learn how to do it safely, is that we could potentially be removing normal organs from a healthy donor that's been whose genetics are well understood and that hasn't been subjected to the pro-inflammatory and cytotoxic effects associated with brain death. We can source those pigs. If we source those pigs from specific pathogen-free facilities, we can be quite sure that they are free of the organisms that we wish to exclude from our recipient, minimizing the infectious risks for not only for our recipient, but also for the rest of the population. Pigs were chosen among domesticated species because they have short gestational period, rapid growth, and are multiparous, so that once we have the right genetics, we can expand that supply substantially and relatively rapidly. And we have, in fact, with xenotransplantation generally, the opportunity to modify both either the donor or the recipient or both, potentially to induce tolerance or at least to reduce the immunosuppressive burden that we need to use to achieve clinical success. The risks are probably well understood to all of you from the conversations that have been going on for the last 6 months since the kidney transplants at NYU and Birmingham and the heart transplant at Maryland. Fundamentally, based on our preclinical work in non-human primates, we think we know what's going to happen when we translate pig organs into man, but those preclinical models have not yet been rigorously tested. And quite honestly, we don't know what's going to happen when we go into the clinic. The endogenous retrovirus issue is one that's been discussed in great detail and that Jay Fishman will touch on in his talk. And finally, once we figure out how to do this, how do we equitably and ethically move this into the clinic on a large scale? So what are the keys to the progress that led to Mr. Bennett's case in Maryland? Well, over the past 2 decades, multiple genetic modifications have been made to the pigs based on some mechanism, our understanding of the mechanisms that lead to pig organ damage when perfused with human blood or when transplanted into non-human primates. And I'm going to focus the rest of my talk on those aspects. And later on, you're going to hear from Paolo Brenner about organ preservation advances, which have truly been a very important key contribution to getting us into the clinic, and then work by many of those on the panel and on the call have contributed to identifying safe and effective immunosuppression as an approach to protect xenografts. And it turns out that's different than what's we currently use for allografts. And finally, again, as Jay will touch on, the infectious disease issues. So how did we decide to make genetic modifications to the pig? Well, the understanding of the mechanisms, the several mechanisms that contribute to xenograft injury are illustrated on this slide. First are the GAL knockout, the antibodies against the GAL antigen or against other either carbohydrate or protein non-GAL antigens. Those then trigger complement activation, depicted as the brown triangles. The combination of the complement activation and cell-mediated binding to the FC portions of the antibodies bound to the endothelium, those things together trigger inflammation of the endothelium, shown here as the red inflammation of the endothelial cell. That by itself is generally sufficient to trigger the phenomenon of hyperacute rejection, which was the initial barrier identified. Once those issues, under circumstances where those issues are addressed one way or another, it became clear that there are additional problems that are unveiled. Incompatibilities between the coagulation cascade molecules, inefficient protection from inflammation, human clotting factors interact with pig protective mechanisms, and then the definition of self non-self that are recognized by monocytes through the CD47 pathway or by NK cells through the absence of HLA-G, HLA-E, for example. So what have we done to modify our pigs in response to this? We first worked on removing the carbohydrate targets. GAL, which is the target of 85% of the anti-pig antibodies in most of us, was the first one knocked out. That unveiled the importance of the CMAH or NU5GC CMAH gene, the NU5GC protein, and finally, Jerry Byrne and Chris McGregor identified the beta-4 GAL as an additional important antigen. With those three carbohydrates removed from the pig is then effectively humanized with respect to carbohydrate antigens as best we currently understand it. There will, in some humans, be antibodies reactive to other things on pigs, whether the swine leukocyte antigens or other proteins. And the notion and to protect against those antibodies, either preformed or if they are elicited after the transplant, that we have added a complement regulatory. Then, if complement is fixed by antibody bound to the endothelial cell surface, the complement regulatory proteins will disable the complement cascade at one of several points in the cascade and will help to protect the endothelial cell from those antibodies. The other two pathways that have been focused on with the 10-gene pig used for Mr. Bennett's heart transplant included thrombomodulin and endothelial protein C receptor. Those work together to more efficiently downregulate the effects of activated human thrombin. And because they communicate through and activate the PAR1 receptor, they trigger endothelial protective mechanisms, which I think are quite important to the successful result that was achieved. Finally, several groups have shown that expression of human CD47 and HLA-E are efficient to protect against monocyte and NK cell activation in the xenogenetic setting. It's hard to test each one of these individually. We have had an opportunity to do some of that in the lung model, but it is not practical to do so in large animal models. I'd like to close by pointing out that the tremendous progress was made with relatively simple genetics. This is from Muhammad from 2016, showing that GAL knockout with one complement regulatory protein and one thromboregulatory protein, thrombomodulin, was sufficient with a relatively simple immunosuppressive regimen to protect grafts quite consistently and reliably. And for as long as the anti-CD40 was continued, these hearts were protected from immune injury and from coagulation pathway dysregulation. This then was the foundation for the German study, which we'll hear about later from Paolo. Which is illustrated in this slide with 4 out of 5 success in an orthotopic model. And this truly was the foundation for the Bennett experiment. The key contribution that I mentioned earlier, and which Paolo will talk about next, relates to the ischemia minimization, which they introduced to the field and which enabled this tremendous progress. I'd like to stop there. Thank you for your attention and be happy to field questions at the end. Thank you.
Thank you very much, Dr. Pearson. That was an amazing insight into your discoveries and progress. Our next speaker is going to be Dr. Paolo Brenner from the Ludwig-Maximilians University in Munich in Germany. He's professor of cardiac surgery and the head of the cardiac xenotransplant program there. Dr. Brenner, thank you for joining us today.
Thank you very much for your invitation. And today I'll talk about organ preservation of xenografts. Is the solution, and surgical aspects of the cardiac xenotransplantation in different baboon models. And there are 3: the heterotopic abdominal, not life-supporting model of Ono and Lindsay, in which the heart must not work. In contrast to that, a life-supporting heterotopic thoracic model of Barnard and Lostman, in which the and great vessels. And last, this gold standard today is the autotopic model of Lower and Chamry, which is used in the behavioral technique also in the clinic. And the first successful results in with HDAF transgenic autotopic xenotransplantation were done in Cambridge with a survival up to 39 days, and we could reproduce it 1998 with a survival of 20, later 25 days, but had a lot of perioperative xenograft failure. And also later, Chris McGregor's group in the Meyer clinic had, after 2000, best results with a survival up to 57 days, but reported over a great number, 50% of perioperative xenograft failure. And he defined it as the so-called perioperative cardiac xenograft dysfunction. This perioperative cardiac output is seems to be induced by ischemia reperfusion injury, and for this is an ischemic crystalloid catapulted Brettschneider or is caused that. And the question is if this stunning myocardium is reversible and could be cleared in our heterotopic thoracic model, which we introduced to prevent this xenograft failure. And here both hearts are working in parallel in our backup system to each other, and the own heart can take over function if xenograft is in dysfunction, is fibrillating, is rejected until to recovery or retransplantation. At that time, we had an old-fashioned immunosuppression with mycophenolate, IVIG, and bortezomib, and so we had 3 cases of delayed xenograft rejection and a survival up to 50 days. But here we had no more xenograft dysfunction, and in this model is limited by a rapidly growing pig heart in the small thorax of an adult baboon. And Jan Arbit could find out that there's also a low pressure curve in the xenograft in the first 2 days. And so this xenograft dysfunction, in contrast to the normal pressure in the aorta, disappears after 2 days, so that we can say xenograft dysfunction is reversible and is not relevant in the heterotopic thoracic model because of the backup system. But in the autotopic model, it has a lethality of about 50%. And then the great breakthrough was made by Mohammed Muyuddin, who found out that with special 2C10 clones of CD40 co-stimulation blockade with a high dosage and survival up to 3 years could be in maximum achieved with 1 year in mean. And we wanted then to reproduce that with the same CD40 antibody and the same triple transgenic donors in our autotopic model in 3 groups. The first one with the crystalloid Brettschneider quadriplegia, and to in fear of a xenograft dysfunction. In the second group, we introduced a new non-ischemic cold perfusion technique from 16. And in the last group, the problem of pig heart growth hypertrophy should be treated with anti-proliferative drugs. And the standard immunosuppression was induction therapies with rituximab, ATT, and the CD40 antibody, versus a maintenance therapy with these antibodies. With a reduced standard immunosuppression, we had no tacrolimus, only MMF, and in the last group, the mTOR inhibitor temsirolimus. To prevent inflammation, additional anti-inflammatory drugs like IL-6, IL-1 antagonists, TNF-alpha inhibitors, and C1 esterase inhibitors were given to the baboons. And so in the first group, we had 3 cases of xenograft failure, and this high troponin level is a sign of ischemia reperfusion injury. And so we started a cooperation with Stig in Lund, who recommended us to cold perfuse pig hearts with oxygenated blood before transplantation. And here we were in a training in the Igelöser farm in Lund with this small heart lung machine of Stig. The stain solution itself consisted of a 8-degree cold kind of Krebs Henselite solution with hyperoncotic albumin, oxygenated erythrocytes, and the heart is constantly perfused during the explantation storage time and also intermittently during the implantation with this self-de-airing cannula. With this small heart lung machine, it was a prototype. And in future, we plan to transport pig hearts over several thousand kilometers to European implant centers, because Steen could also show that after 24 hours of cold perfusion, you can successfully transplant pig hearts autotopically. And so in Sweden, a clinical study was started, and in the moment, a multi-center study also in Germany is ongoing. So in the second group, we had no more xenograft dysfunction with low troponin level. But here we found massive hypertrophy and growth after 40 days with about 200%, which is not only natural growth, but also hypertrophy, because blood pressures in baboons are higher, 120 to 80, in contrast to piglets, 80 to 40. And so we solved this with antihypertensive drugs and early steroid reduction, like in children heart transplantation. Steroids make also hypertrophy, and with the mTOR inhibitor, the rapamycin derivate temsirolimus. And so we could finish our 3 groups. The first Brettschneider group with xenograft dysfunction, and the second without growth control, with diastolic dysfunction, liver congestion, and with growth control, 6 baboons could achieve the end of study. 90 days had to be terminated according to the guidelines of our government, but 2 could be prolonged to half a year, and 2 died of an PCMB positive donor with sepsis and multi-organ failure. So that was detrimental. And that was then also, here's a survival curve, then published in Nature in December 2018. Later, the completed 6 of 10 study in the ISHLT journal is a great breakthrough. And in hemodynamic data, stroke volume index and cardiac index is significantly increased with cold perfusion in these 2 groups, and also catecholamine dose in duration is minimal after cold perfusion. And the pig heart increases in mass after growth control only by 22% after 80 days, and without growth control, it shows this diastolic dysfunction. And also after half a year, there's only a minimal increase of left ventricular mass in echo. But if you stop the mTOR inhibitor, there's a sudden re-increase. So that in future, we will solve this problem of pig heart overgrowth with growth hormone knockout pigs, which are smaller and have smaller hearts. And in parallel, we also breed Auckland Island pigs from New Zealand, which are free from Perf C and hepatitis E, and have an ideal body weight of 80 to 90 kilograms, which is perfect for patient, but also for baboon experiment. And they have also been in a clinical trial with Bob Elliott's islets 15 years before. And here I compare our data with some from Baltimore. Here, Moyuddin and I reviewed it for a Frontier of Immunology, compared the crystallite solution with stain solution and another blood cardioplagia, Del Nido, which I also use in Munich since 2 years in bypass surgery. And here we found a xenograft dysfunction with crystallite solution in 62% detrimental effect, and only in 33% with the Del Nido solution, and only 20% the best with steam solution. Here's the greatest survival curve of the other topics. And so the question exists, must also other blood cardioplegic solutions like Heidelberg or Calafiori should be tested for xenotransplantation? And in the last paper, Moyuddin showed his best results with multitransgenic and the survival up to 264 days with growth hormone knockout, and without growth hormone knockout, that was this survival. And the worst results were without thrombomodulin. And here you have found thrombi in the right and left ventricle, so the thrombomodulin is very essential. And in the fourth group, there was only minimal complement and immunoglobulins. And so the cold perfusion was then also used in the highlight of this year, the first in man cardiac xenotransplantation from 7th of January with compassionate use in the 10-fold genetic modified pig with cold perfusion and the growth hormone receptor knockout, and details will be showed from Moyuddin after that. And so where are we now? We could show that this perioperative xenograft dysfunction was also found in the heterotopic thoracic model, but here it's not relevant because you have a backup system, and it's a reversible cardiac output process, a stunning Meyer card, and can be caused by the ischemic crystalloid cardioplegic solution, for which xenotransplantation is not ideal. And we could successfully prevent it with the cold perfusion technique of 16. We could show that co-stimulation blockade works in the orthotopic model, and together with rituximab could prevent hyperacute and delayed xenograft rejection. And the picard overgrowth was inhibited by anti-proliferative drugs, so that the preclinical study was now completed in 6 of 10 baboons living up to half a year. And so the ISHLTv guidelines for first clinical trials were now fulfilled. And one spin-off is that also a cold perfusion with this new white boxes in the clinic in allotransplantation. And I did the last heart transplantation 6 weeks before without catecholamines with a patient who was standing beside the bed the morning after operation. And what must be still done? We should also test other blood cardioplegic solutions like Heidelberg, Calafiori. We should use mini pigs on a growth hormone knockout on Auckland Island, a background, and to test other humanized CD40 and ligand antibodies, and can now also extend survival in our baboons in Germany up to 1 year. We must have pathogen-free donors, which are free from PERF, PERF-C, and porcine CMV, which are very important. We must have additional knockouts, new 5GC beta 1 for gall. And also in Germany, now an approval procedure for cardiac xenotransplantation as a clinical therapy is necessary. We do with the Paul-Ehrlich Institute. And in parallel, the planning of a clinical phase 1 study with involvement of the ITI committee is now ongoing. Thanks to all our collaboration partners, especially Mohammed Moyuddin in Baltimore and 16 in Lund, and that we were very successful, showed also a lot of prizes we won in the last 3 years. Thank you for your attention. Your microphone.
Thank you very much, Dr. Brenner, for your presentation and with the awesome insights into xenotransplantation. I'm glad to have you at the panel in the end, and I think there will be a lot of questions for you. Our next speaker is Dr. Mohammed Moyuddin. He's professor of surgery and director of the cardiac xenotransplantation program at the University of Maryland, Baltimore in the USA. Dr. Moyuddin, welcome to the program.
Thank you very much, and thank you for this invitation. The task that has been given to me is to talk about how xenoimmunosuppression regimen differ from allo regimen, and why could conventional immunosuppression overcome xenograft rejection. What's the influence of genetic modification on modifying the immunosuppression, and what role growth hormone receptor plays. What is an optimal immunosuppression for xeno, and the lessons learned in regards to immunosuppression from Mr. Bennett. So for allotransplantation, I mean, unless you have some indication for induction therapy, those are highlighted here, and I won't go into the details. You know, a standard therapy of immunosuppression is used, which includes tacrolimus, mycophenolate, mofetil, and prednisolone. However, xenograft rejection is a very complex phenomenon, as Robin alluded to in detail, and you see hyperacute rejection within minutes with the antibodies, the pre-existing antibodies and elicited antibodies against carbohydrate antigens on the pig endothelial cells, leading to microvascular thrombosis and myocyte death. There has been several attempts to use conventional immunosuppression to overcome this xenograft rejection. Dr. McGregor has shown both in heterotop models on the left that he can get up to 137 days survival using induction with rituxan and thymoglobulin, and then treating these baboons with tacrolimus, sirolimus, and steroids, and got 137 days of survival. And in his orthotopic model, he got about 57 days of survival using the same regimen and using gall knockouts and CD46 transgenic animals. And this slide, Robin also showed that conventional immunosuppressions have found to be less effective in the xenotransplantation model, and initially the CD154 based regimen, were successful in prolonging the grafts better than the conventional regimen. However, the thromboembolic complications of CD154, precluded its use in the future experiments. Here, Dr. Cooper and his group tested different combinations of conventional immunosuppression, and was shown that that they were not effective in prolonging the grafts, as seen in the red line here, versus when he used anti-CD40 antibody in his regimen along with either rapamycin or MMF. So this is just a summary of those results, and as you can see that the conventional immunosuppression in heterotop model only got survival up to 137 days, and we have shown in our experiments that in the heterotopic model we have gotten survival for up to 945 days using 3 gene picks which have CD46 and thrombomodulin. So this has become like our standard regimen since then, and which includes the induction with ATG, rituxan, 4 doses of rituxan, and complement inhibition initially with scopolamine factor, and lately we've been using C1 esterase inhibitors. Our maintenance have always been since that time with anti-CD40 antibody at a higher dose, 50 milligrams per kilogram. Along with it, we use MMF continuously throughout, and then we taper down the steroids over 2 months. We also give in our heterotop and orthotop models of non-human primates heparin infusion, and also ganciclovir to prevent PCMV infection. So this was what Paulo also showed. Our main paper that's showed survival of over 900 days. It showed the critical role of anti-CD40, because when we reduced the dose earlier, within 100 days, all the grafts rejected. But when we reduced the dose after 100 days, even reducing to half did not cause rejection. Only when we completely stopped the antibody and the antibody was removed from the circulation, all these grafts were rejected, and they were all rejected because of the antibodies that came back with a vengeance. As you can see here, the time points where the CD40 therapy was stopped, as shown with the red stop signs. And on the right, you can see that the circulating anti-CD40 was removed, and when at the time point 2 months after, when it was completely removed, these grafts rejected, and they all showed antibody-mediated rejection pattern. So not only the immunosuppression, I mean, the genetic modifications have helped, as you can see here. The therapy, when used with gal knockout and CD46, did not prolong the graft, but when the thrombomodulin was added, we started getting significant graft survival. So with that proof of concept, we moved to orthotopic model, and as Dr. McGregor described, we encountered the PCXD initially, and all of our animals were going through graft failure within 48 hours time period, and with the description and the publication of Dr. Bruno Reichert, Paulo Eckert's group, the preservation method that they described, changed the scenario for us also, and we started using this gene solution as described by Paulo just a few minutes ago, and since then we have avoided the PCXD in all our animals, and all our animals have survived beyond that period. In the meantime, we've been testing different gene modifications, and these pigs produced by Ravi Vikor, one of our close collaborators. So the recent pigs that we are being using, and which was used in Mr. Bennett, we had 10 gene modification with 4 knockouts, 3 carbohydrate knockouts, and also growth hormone receptor knockout. And it also had 2 complement regulatory the transgenes and 2 thromboregulatory transgenes, and also 2 anti-inflammatory transgenes in it. So with that, modification, we have progressively shown, improvement of graft survival. The red one is the red line shows the graft survival of the group that was not 10 gene, but had this group did not have SEMA, but had the growth hormone knockout. However, when we use the 10 genes shown in green, our survival was not as good as the ones without SEMA. So as you guys know, that there's non-human primates do have SEMA gene. That's why they when we remove that, there are publications showing that they reveal another neoantigen, and that can produce more antibodies. So there may be a reason why the survival was less than our 7 gene pigs. So as Paulo showed, the growth was one of the things that we considered based on their publications, as shown here. They help the growth by using temsirolimus, which restricted the growth, and when it was removed, the growth started to happen again. But in our case, we had we used growth hormone receptor knockout pigs, and as shown here on top left, the growth hormone intact pig and the growth hormone negative pigs. However, when we looked at the histology, we saw rejection being a significant part of this growth, as shown here in the histology, where we see typical, rejection pattern with microvascular thrombosis, edema, and intervascular hemorrhage, and all. Whereas in the animals without growth hormone, we didn't see any such the pathology. So, we have a hypothesis that is a multifactorial process, and it requires both extrinsic factors, which Paula described. Rejection plays maybe a significant factor, and there are some intrinsic factors that also involved. So here I'm just showing some of the modification that we are doing to avoid some of the rejection pattern, and what clinically available drugs are available, and then can be combined with these modifications to overcome xenograft rejection. With that, I'll move to our first in man pig heart transplant experiment with Dr. Griffith and I approached the FDA and had and were able to do this first in-human transplant on January 7th. So the immunosuppression regimen had to be changed significantly in this patient. Our non-human primate regimen is shown on the right, and our the regimen that was used in Mr. Bennett is shown on the left, which was again based on CD40 monoclonal antibody treatment. However, the B cell depletion was not as robust as we have done in our baboon, and also because of his leukopenia and thrombocytopenia, we started ATG after the transplant instead of giving before. C1 esterase inhibitors were also given, and a smaller dose of MMF was also given, which was held between grade 20 and 50. And we also used the tapering dose of steroids in this patient. So just to show you that, even with this minimal therapy, we were able to, on the top panel, where you can see that we were able to deplete B cells effectively from the periphery. However, in the middle panel, as you can see, the plasma cells were still intact because, as you know, the CD20 is not expressed on plasma cells, and they are not depleted with it. And on the bottom panel, 2 days later, when we took that lymph node out and looked at the B and T cell population, we saw a significant amount of B and T cells, showing that our depletion strategy was not at par, and so the question arises: how much is needed to deplete the B cells in the lymphoid organs? So here we are showing that that the antibody production against pig cells was also suppressed over a long period of time until the day 40, and you can see 2 peaks at that time. But now we also considering we had to give IVIG because his immunoglobulin levels were low, and maybe what we are seeing here is the immunoglobulin levels. Because here you can see predominantly IgG was higher. So with this, I mean, what are the factors affecting IS in this patient? We think the pre-existing conditions is thrombocytopenia, leukopenia, and being on ECMO for 40 days before transplant definitely affected it. The patient had type A dissection, which led to later on, possibly bowel ischemia and an effect on renal function. His immunosuppression was not continuous because we had to stop MMF for 30 days due to very low WBC count. We had to administer IVIG at one time because his immunoglobulin levels were very low. He was on renal replacement therapy. And then infection, we have found that the PCMV DNA in the blood without any evidence of active CMV infection or viral replication on either histology, electron microscopy. And then, we cannot disregard atypical rejection because this heart was well protected by several gene modifications. So I mean, lastly, I mean, there are 2 questions, we have to balance between the immunosuppression and gene modification. And somebody asked the question in chat that, if we can overcome completely the immunosuppression by gene modification, maybe at one point, or by inducing tolerance, we can overcome this. And then you have to be very careful with immunosuppression, whether, like in this case, that we had to deal with, whether to fight infection or to save the heart by giving more immunosuppression. So that was another critical decision that we had to make. So in the end, I think that this was a huge success because we didn't know that this patient will survive the operation or how many days he'll survive. He survived for 60 days, and it was his courage that helped him, survive for this long. And I'm very thankful to Dr. Griffith also that to providing this opportunity for taking this this process from bench to bedside. And this is our team that took part, and I'm thankful to many people for this procedure. Thank you very much.
Thank you so much, Dr. Mohiuddin, for sharing your insight there and also your most recent experience. This is a perfect segue to our infectious disease topic that we will have next. And our next speaker is Dr. Jay Fishman, Professor of Medicine at Harvard Medical School and Director of the Transplant Infectious Diseases and Compromised Host Program at Massachusetts General Hospital. Thank you for joining us, Dr. Fishman.
Well, thanks for inviting me, and thanks for allowing me to join my friends and colleagues in this discussion. I was given a slightly different challenge than some of the others, which was, what have we learned coming out of, or almost coming out of, our SARS-CoV-2 pandemic? Because some of the questions related to infectious disease are actually quite similar. What I would say is this: what do we do when confronted with a potential pathogen? Well, such as SARS-CoV-2, we need new assays. We need to understand the biology of those organisms. We need to think about ways of preventing disease, so vaccination or antibodies or new antiviral agents. We have to think about our special hosts, our transplant population, other immunocompromised hosts, and think about the ethics. Are we giving equal access to all groups, to all therapies and all opportunities? And although it sounds naive, I would say politics should have no role in these kinds of discussions. So what I'm going to try to do today is share what I've learned over 20 years of thinking about transplantation in terms of infectious disease. Which organisms are going to be common and are going to be challenges for our patients? How do we develop pig herds that are safe for use as source animals for xenotransplantation? And which organisms should we think about based on our experience in allotransplantation? How do we deploy the various diagnostic tests to optimize safety for the patient and for people that surround the patient? And how much risk is acceptable? And the take-home message is fairly straightforward. We have to define acceptable parameters for safety in clinical xenotransplantation. Can we make it as safe or even safer in some ways than allotransplantation? Can we identify potential human pathogens? And we focus on viruses because they're the most difficult both to detect and to treat based on past experience, and then to develop assays and preventative strategies for those. And we must extend knowledge. We have to develop the science of these potential pathogens in clinically relevant models such as immunocompromised hosts in vivo. As you all probably know, there's this nexus between epidemiology and the net state of immune suppression, what the individual is exposed to as a transplant recipient, including from the organ donor, and all the things that go into infectious risks in the recipient, including how healthy are they, and is their immune system working at baseline, and have they been vaccinated against various pathogens. In xenotransplantation and in allotransplantation, we bypass host defenses. So we implant an organ directly into the system, and potentially, as you've just heard from Mohammed, we may need increased intensity of immunosuppression. The graft itself provides an ecologic niche for pig-specific organisms, porcine cytomegalovirus, for example, and the major histocompatibility focus dissonance may protect the graft from normal immune function. We may not have the right assays, and we may not even know which organisms to look for, and genetic modifications of the donor may change the nature of the organisms that we see. So what we want to do is make a list. We want to make a list of organisms that we don't want to see in our recipients, and therefore we exclude to the degree possible from our donor swine. Can the organism cause important infection in the swine or productive infection in normal human cells in vivo? Can it cause persistent infection? Can it change the behavior of other organisms or recombine with host DNA? Are preclinical xenograft models, such as primates, even relevant? For example, porcine endogenous retrovirus doesn't replicate well in most non-human primates. Could we expect spread to the general public, and do we have the right assays? And years ago, I divided various pathogens up into groups based on whether or not we had tests and whether or not we had therapies. So common human pathogens, such as Epstein-Barr virus or cytomegalovirus or pneumocystis, we have good tests and we have good therapies. There are some common zoonoses, organisms from animals, that we might expect could be in donor animals, such as toxoplasmosis or leishmaniasis. And again, we have adequate tests and reasonable therapies. There are species-specific organisms, such as porcine cytomegalovirus, where the assays aren't quite as good. Some of them are laboratory research-based, and there are a few validated assays for use in humans as well as in breeding colonies. There are some organisms that we don't know very much about, but we know that similar organisms cause disease in immunosuppressed human recipients. Adenovirus was or the mycobacteria in that setting. And so we may have some tests, but we really don't know very much about therapy. And then the unknown pathogens such as porcine endogenous retrovirus. Now, amongst the organisms that we've studied, porcine cytomegalovirus, for example, causes disease in the graft itself and can cause endothelial activation with coagulopathy, consumption of white cells, consumption of platelets, and promote graft rejection. It can be removed from the colony by careful breeding, but is easily reintroduced, but can't really be detected unless you immunosuppress the donor animal. The porcine lymphotropic virus doesn't seem to infect either primates or humans. Circovirus causes pneumonitis in swine, but isn't really a problem for humans. I'll talk a little bit more in a second about the porcine endogenous retrovirus, which causes no infection in swine, but doesn't cause productive infection in baboons, so that they're not a very good model for studying it. Humans carry the receptor, but has unknown virulence for humans. And we haven't ever seen replication in humans, even when exposed to porcine endogenous retrovirus containing organisms. And we do have good antivirals from our experience with HIV that work against PERV in vitro. And then the coronaviruses, pig and human coronaviruses, don't appear to cross species. So I put together a list of organisms that I would prefer not be in donor swine, but the goal is to make sure they're excluded, not necessarily from an entire herd, but from the organ that we're giving to a human recipient. I said I'd come back to talking about the porcine endogenous retrovirus, and my lab cloned the virus in 1997 after knowing that a virus appeared to exist in transformed cells and lymphoma cells of swine. And we cloned this from normal cells from our pigs. This was PERV-C. And then subsequently, others identified PERV-A and B, which were both infectious for human cells, and that we could replicate those experiences in vitro. As I say now, there are 3 types of PERV that differ in various regions, and PERV receptors are present on human cells. But productive infection of normal human cells has not been demonstrated. So all the work on PERV has been using a permissive transformed human cell line that lacks certain endogenous factors that prevent replication. Replication efficiency improves with passage in vitro and produces a recombinant. So you may hear about PERV-AC, which is a combination of PERV-C and PERV-A, which reproduces better in vitro, but still has not been shown to infect normal human cells and has not been demonstrated in prior exposures to pig tissues. You may know that a number of years ago, we participated in studies that show that using CRISPR-Cas9, we are able to knock out all copies of PERV, A, B, and C, from a cell line that produces large numbers of PERV elements. And therefore, genome editing allowed not just the introduction or deletion of genes related to immunology, but also to infectious disease. And so in thinking about the risks of infection in human recipients of pig organs, we have a combination of two timelines. And you may have heard me talk in the past about the timeline of transplant infectious disease, where over time, that risk goes down, but initially you have nosocomial exposures and exposure from the donor organ. And then as the effects of immunosuppression accumulate, we see what we call opportunistic infections, organisms such as pneumocystis and toxoplasmosis, the hepatitis viruses and the herpes viruses. And as we taper immunosuppression over time, those risks seem to go down. In addition to that timeline, we now have a timeline for pig-specific pathogens, which we won't really be able to clarify until we do clinical trials. And so that we know that there is some additive risk from the pig donor organ, which will be determined by vaccination, prophylaxis, and the breeding of swine, and will allow us to address these two timelines together. We do know that in relation to what I said at the beginning, we need good assays for pig organisms, both in swine, in pre-human studies in primates, and in humans. We know that serologic testing is the most useful for looking at past exposures, and that routine cultures may or may not be available for many pig pathogens. And so Hartline and colleagues developed multiplex PCR assays similar to the ones we developed in my lab to show that we can detect activation of various organisms in the setting of immunosuppression that may not have been detected in the immunologically normal host. If we have an individual who has an infectious syndrome where we can't detect specific pathogens, we may apply metagenomics, whole genome sequencing, to look for novel pathogens derived from either the host or the donor or from the environment, which may infect immunosuppressed hosts. And this is something that we're doing clinically right now for our patients. And then we have other types of assays to look at B and T cell function and to look at other infectious exposures. So these assays need to be validated further. How do we enhance safety in clinical xenotransplantation? Well, I've already mentioned we need to develop assays for pig-specific pathogens and then appropriate preventive strategies for each. We need to characterize the biology of these organisms in immunosuppressed hosts, but if there are no receptors, we will have to wait until the clinical trials to advance our knowledge in the risks of infection in xenotransplantation. But we do have good antiviral agents, antibacterial, and antifungal agents for use in those recipients. We have to avoid subjects that have pre-existing immune deficits or active infections, just as we do in allotransplantation. We will archive blood and tissue samples from the donor animals, as well as from the recipient and perhaps from surgical teams to use in investigations should infection emerge during the course of clinical trials. We will monitor our recipients for routine and pig-derived infections, and we have to recognize, as in allotransplantation, that some risk for infection will always occur. Some risk for potential malignancy will also exist, just as they do in our allotransplant recipients. And we need to develop protocols so we know what we will be doing when these patients arrive to be taken care of, but it's driven by our experience in allotransplantation. So we will deploy assays for our source animals, for monitoring our xenograft recipients, for looking at individuals who have infectious syndromes, and for some healthy contacts of our recipients in society. The infectious risk of any immunocompromising condition can never be zero. We will always have some risk of opportunistic infection, whether it comes from the environment, the donor, or the recipient. Common human pathogens can be expected, and we may need to use new technologies such as whole genome sequencing to elucidate the risk of infection over time. We will archive donor and recipient specimens for epidemiologic studies, but we will only understand the risk when data from clinical trials emerge. I should point out that these organs from swine are resistant, to the best of our knowledge, to hepatitis viruses and to HIV, and likely to other common human pathogens. So there may be advantages for some of our recipients in terms of using pig organs over human organs. That doesn't get talked about very much, in addition to the global increase in the number of organs. So thank you very much for including me, and I'll stop there and entertain questions at the end with the others. Thanks very much for inviting me.
Thank you very much, Dr. Fisher, for this very interesting presentation. So we will have you two at the end of the session in the panel for questions later. So our final speaker is Dr. Richard Chinock, Chief of Medical Office, Loma Linda University Children's Hospital in California, USA. Dr. Chinock, thank you very much for joining us today.
There we go. It seemed like it wanted to get to full screen. Thank you so much. This is quite an honor. The prior panelists have taken care of some of my work, which I anticipated a little bit. But I know I'm presenting on behalf of Dr. Bailey and the team at Loma Linda. I'm sorry that he's no longer with us. We just had remembrance of 3 years since his passing. And I know that he would do this with more elegance and grace. And I know there's no way to cover this adequately in 10 minutes, but what I plan to do is just give a hint of a context for baby Faye and ask questions regarding the ethics of xenotransplantation that I think we should all consider. So I'm a big believer in starting with the why. And though it's old and grainy, and I hope it plays properly, let me start with a clip of Dr. Bailey describing his why.
About 10 years ago, when I was in training at the Hospital for Sick Children in Toronto, Ontario, Canada, I became deeply impressed with the fact that we'd seen a number of babies with incurable heart disease, incorrectable heart disease, babies in whom you could only experiment surgically with. And it impressed me that what those babies needed really was heart replacement.
So I just got to tell you how fortunate I was to work with a man who starts a journal article with "Bless the Babies." You know, this was his passion, and he felt at the time the only option was xenotransplantation. Obviously, it's progressed from there, but with this context in mind, I want to begin the conversation regarding xenotransplant ethics. And while I have thought a lot about this, I'm not a trained ethicist, and so I won't be delving into utilitarianism and all those other isms that most ethicists will do. I'm just a simple country pediatrician, and my framework, there are basically two questions. Can it be done? Should it be done? So the previous speakers have elegantly described the challenges of making this clinically meaningful therapeutic option. So I'll spend more of my time on the should it be done. Others have talked about immunologic barriers and the ability to provide sufficient animals, so I won't go there. But in should it be done, I think the questions that need to be asked is can we create useful longevity? Is it destination or bridge? Infectious barriers, Dr. Fishman has just really elegantly handled. And then I think there's patient, family, and social perspectives that are important as we talk about this. It's very interesting as I was rereading the editorials and commentaries after Baby Faye that there were three main criticisms of the consent process, and I think that has implications for how we approach the ethics. The first was they said there was no mention of looking for suitable infant donors after the baboon was implanted. They said we should not have transplanted across the ABO barrier. And the consent also said that long-term survival is a possibility, and they criticized that. So let's just talk about those in just a brief second. If you had a functioning xenograft in place, would you take it out? And I know I had lots of conversations with Dr. Bailey, and he basically said, if it's working, it's not coming out. And using it as a bridge buys time for that particular patient, but it doesn't solve the issue of insufficient human donors, because you're just changing the time from one time to another and still using a donor. The question of ABO incompatibility has been definitively answered by Dr. West, and it's of interest to note that she came to Loma Linda and did a thorough review of the data from Baby Faye and concluded that our explanation of ABO incompatibility as a cause of death was likely flawed. And we in most centers now routinely list all infants as ABO agnostic for the source of their donor. Regarding the notion that long-term survival couldn't be predicted, Dr. Bailey and his team performed 400 transplants in animal models prior to Baby Faye. And the picture on the right is the picture of a mother goat who was transplanted as a baby goat and then grew to maturity and was able to have her own goat, baby goat, there. So in that animal model, he had demonstrated a natural lifespan. Of course, that's several years as opposed to decades, but it still seemed at least reasonable. The other thing is that he had a primate to primate model, and we had a number of baboons that survived beyond 2 years and were eventually euthanized because the research project completed. But it was possible to get beyond the 20 days that Baby Faye survived. Dr. Fishman talked about this, but I think it's important to realize, and this impacted our research project at Loma Linda. This is 1984, right here when Baby Faye was transplanted, and it was right at the beginning of the AIDS epidemic, and there was a lot of concern about retroviruses, and in fact, the FDA put a hold, and that effectively eliminated our research project at the time, and it seemed like much of the world moved on to pigs, and understandably so. So I'd like to spend the last few minutes describing what I think might be the most salient ethical issues and how we address the patient, family, and society concerns. So here's a clip of Baby Faye's mom describing when she first met Dr. Bailey.
I wondered if the doctor was a mad scientist.
So, a scientist, not mad, but that's a common kind of reply that the public would talk about. The context for what was happening at the time, and it still applies, 25% of infants die while waiting. But if you're having a conversation with a family, are they able to have a rational, reasoned decision for xenotransplantation given that statistic? I know for Baby Faye's mom, she said she was willing to do anything, and I know many moms would say the same. But for xenotransplantation, what's the goal? Are we just answering a clinical question, or do we really believe that we're providing a long-lasting benefit to that particular patient? Will a xenotransplant lead to more or less suffering for the patient? And then is the family, or are we prepared for the media scrutiny? And I was searching for pictures of the animal rights protests that happened at Loma Linda during this time, and I couldn't find it. But while the public is generally supportive of transplantation, Xenotransplantation seems to be a different matter. Now, I think, and studies have proven this, that they accept a pig much better than a non-human primate. But I think few people know that for 2 years after baby Faye, Dr. Bailey wore a flak jacket whenever he was out in public, and he had personal security at his home because he had multiple death threats. And it was, you know, the families could also experience some of that in case, as they decide whether to accept it. And then if we provide xenotransplantation as an option before we're ready to do it large scale, will the public say, hey, you can just have a pig, I don't need to worry about donating? Will a family say, I'm not going to donate my loved one's organs? And Dr. Fishman assessed the SARS-CoV-2, but I think people are very sensitized to zoonoses, and it will add some additional challenges to our conversation. Is the public obliged to accept some risk to save a limited number of our fellow citizens? I believe that we need to have an important conversation about that, and maybe before we get to the next xenotransplant rather than after. And we, as a transplant research community, are obliged to do all within our power to limit risk. And Dr. Fishman describes those risks that he was talking about, which I think are the main risks that people would be worried about. Others have touched on healthcare equity, and to me, it goes both ways. Will we give equal access to organs, but will we also give equal risk of research, and not use vulnerable populations for research and have that shared risk amongst us all? Just some final thoughts. Somebody asked in the chat about xenotransplantation in newborns, and Dr. Platt and me and Lori and really are on record as saying that we believe that's a viable option along with others. So I believe that is the best model, but of course that's just a small sliver of what is needed for transplantation. So donor scalps continue to drive the search for alternates, and xenotransplant has a significant potential, I believe, should be pursued. But it requires a lot more work than I think that we have done as a transplant community. And radical honesty with potential recipients is mandatory. And with that, I'll finish. And thank you very much.
Thank you so much, Dr. Chinook, for your very thoughtful addition, and to all of our speakers. We now are fortunate to have an additional 3 very distinguished panelists that will join us. Professor Bruno Reichert is one of the pioneers of xenotransplantation, and he is past president of ISHLT as well as Lifetime Achievement Award recipient. We have our current ISHLT president, Andreas Zuckermann, and cardiac surgeon from Vienna, Austria. And we have Professor Georg Markmann, who is an ethicist and runs an ethical program that specifically has focused on challenges on biotechnology medicine as well. So maybe we can start off. I don't know if everybody can start their videos, and then whoever speaks start their microphones as well. Maybe we can start off with one of the questions that was posed here before, which is, how likely is it that we can modify animals to the point that we don't even need any immune suppression anymore? And I want to add a few things to that question. One is, how ethical is it? What are the limits of what we should be doing? And then also use the opportunity to have all you pioneers around here and maybe share some pearls of things that you may have discovered on the way that were very unexpected. As we know, we often only understand the pathways once we start targeting them with therapies or modifications. Maybe, I don't know, Professor Reichert, if you wanted to start sharing some of the pearls you have experienced along the way and what you think around the limits of manipulation. You're still muted.
Muted now. You can hear me, yeah? It's very interesting, but for those who are not so familiar with xenotransplantation, I would encourage them basically, xenotransplantation is not that difficult as it was just exposed by the various discussions. And it is simple. Our livestock already for the baboon experiments is very healthy, very safe. You need to view a few additional microorganisms. I know that the changes, genetic modifications we have in hands, and it is our policy to keep them as little as possible. Very effective. So doing a lot of human heart transplants, it's always a struggle to get the patients, sometimes to get the patients off the extracorporeal circulation. We do that in Munich now since December 2015, and except for two cases, we could all get the animals off the extracorporeal circulation with a beating heart, with a beautifully beating heart. So it works, and I'm excited about that. And I think we all need to keep it simple, more simpler. Otherwise, it will not work, and it will not get accepted by the regulatory authorities in Europe as well as in America. That's my impression.
I think, Bruno, I think you're right. I think it's going to be possible to succeed with relatively simple genetics, as you have done in the preclinical model. I think the question that came to, is it possible that we can make further modifications that will make life easier for us in the short term or in the long term? I think that may come to pass. I think the first clinical trials will happen with simple genetics and some version of the immunomodulatory treatment that Mohammed and you and we have shown is quite effective based on co-stimulation pathway blockade. That is a pretty simple and non-toxic and effective recipe if you have the genetic protection against dysregulated coagulation, which, as Mohammed said, and as all of those of us who've worked with these organs have found, is quite protective for that otherwise very important problem. So if you wanted to make a pig less immunogenic, you could knock out its swine leukocyte antigens. But as Jay will tell you, if you don't have SLA, the animal may have health problems with defending itself against infection. And that's no guarantee that you're actually going to help yourself by, in the long run, against the human response against the pig. The idea of introducing immunomodulatory genes into the graft is intriguing. It probably will work. But if you find that that immunomodulator that you put into the graft starts leaking into the patient, you could end up with an over-immunosuppressed patient. So anything that you try like this, and I think that's what you're getting at, try to keep it simple, that if you make many different changes, there will be downsides. There will be complications that will show up as a consequence of the additional things that you try to change.
May I ask the experts in the field who are sitting all there, I would say the big three from the big centers, discussing about the phase 1 trial, who would be the patients that you would select to participate in the phase 1 trial? I think this is a very important question, ethically, but also selection. Definitely, we know Mr. Bennett was not the perfect transplant candidate, but who would be the patients that you would select for getting into a phase 1 trial?
Yes, I'm asked, shall I say something? Okay. Well, as you said, Mr. Bennett, I'm completely respectful. I'm not criticizing Mohammed. I'm not criticizing Bart. I don't want to be misinterpreted. And it is not my policy to say anything bad in front of such a huge crowd. I would never do that. But I know they were under pressure. FDA put a lot of pressure on them. Which patient, how the patient should look like. And one cannot argue that Mr. Bennett was unbelievably sick. But it doesn't, the sentence doesn't help to say he would have died anyhow as well. I think we need success with our first patients, long-term success. So a patient of 41 days on an extracorporeal oxygen on an ECMO is just not an ideal patient. So I think a patient on a mechanical assist device other than an ECMO would be much better. And what was not that by Muhammad, they did an unbelievable job, but at the end the heart was beating nicely, what I heard. I mean, the porcine heart after surgery in early early January, but when they did an echo, they found out they had a type A dissection all the way down of the aorta. Yeah, that's in such a situation an almost unsurmountable complication, and it couldn't happen because the patient was too sick, too long on the ECMO, and so on. Yeah? So again, very respectful, and it compromised the renal artery, compromised obviously the mesenteric artery, and so the patient had obviously bacteria from the bowel in the blood. So that means that it's a sepsis, and sepsis in a healthy patient has a 50% mortality. So there shouldn't be any criticism about that. So it was an utmost difficult patient, and with a full-blown complication, and nobody is actually did a bad job. Actually, Bob Griffiths did a beautiful job to get out of that water hole. And so we need better patients. And to answer your question is, and not to overdo it now, that we must do this and that and look at that. An unknown organism that makes you crazy, you know. And so start again thinking simple. Now, to answer your question would be, there are plenty of patients, they're up for a mechanical assist device, but they can't have it because they have a bilateral heart failure, left heart failure, right heart failure. And there are plenty of patients who don't like the idea to be on a drive line, to have a mechanical assist. So they have mental problems to be on a mechanical assist device. So there are plenty of patients, or you need two anticoagulants. So if you had maybe an ulcer or something like that, a bleeding problem, you can't do that. So that they just bleed to death if you put in a mechanical assist device with two anticoagulants. So there are plenty of patients, first patient, good patients, very sick patients for a porcine heart transplant. So I would use these patients, and please don't operate on them unnecessarily before you do that P-card. The P-card is working beautifully. Yeah.
That was a good summary. I would add that patients with hypertrophic cardiomyopathy are poorly supported by our ventricular assist device options, and they might be one category. Another large category is patients who are highly sensitized against human antigens and who are otherwise a wonderful allograft recipient candidate, but either the desensitization process is quite toxic. And as long as that patient does not have antibodies against the pig, that might be an ideal population of patients to take on early on. So there was a question in the chat about pediatric infant heart transplants. It is conceivable that that will turn out to be a work, I think, a little bit more proof of principle should go on in the laboratory before we take that step, but it is possible that that might have immunologic advantages. So, Mohammed, did you want to answer too?
Great. Yeah, I think Dr. Brenner wanted to chime in there as well.
I think the best patient would be, let's say, 67 years old. In Germany, over 65 years, you don't get an allograft. You must have a bilateral heart failure, and you normally get a Berlin heart. A Berlin heart, you must stay in hospital, and it's only for bridging. So older than 65, not on ECMO, not with an assistive valve, no redo operation, that would be ideal. And this diabetes is exclusion criteria for allotransplantation, and allosensitized also. That's a typical patient you can begin in the pilot study. And eventually you can make a bridging also under 65 with Professor Reichert told a patient who could not get an LVAD because they have stomach bleeding and so could not have anticoagulation. That was a typical patient for xenotransplantation.
And what do you think is the timeline for start of a trial?
Timeline, Bruno, we want?
Yeah, I think in 2 years' time, I think we are in the end game, and we are testing a clinically acceptable antibody, and we need smaller animals, and we need herd animals from a herd, not clones. And I think the ideal would be a small species like the Auckland Island pigs we have. The sows will grow up to 70 kilograms, and the boars to 90 kilograms would be ideal. So to avoid these overgrowth problems, it's inherited. So pig hearts still think they are in a pig and not in a baboon, and not in the future in a human. Yeah? And a German landrace pig grows up to 300 kilograms, and the heart is weighing a kilogram. Yeah? And actually, if you listened to the talk from Loma Linda to Dr. Chenok, and if you looked at that goat, yeah, and so Leonard Bailey, whom I have known, had the honor to know him, and I admire him a lot, you know. And if you transplanted a goat heart when the goat is 4 kilograms, it reaches, I think, 70, 80, or 100 kilograms after a year, and the heart grows. And the heart grows not because there's a demand, there's a bigger body, because the heart genetically knows it has to grow and has a certain size after 1 year. Yeah? And the same is, I've done plenty of pediatric heart transplants. And you never have to retransplant such a heart. It will just grow because the human heart just grows. And it knows how it must be at the age of 18 years, after the adolescent stops. So growth is important.
Okay, unfortunately, we're already over time. However, I want to give Dr. Markman the chance to chime in a little bit from an ethical perspective. Please also use the opportunity to just contact, I think all of our speakers are probably on the ISHLT Connect platform, and you can find their emails there. So please use the opportunity to also directly email them and ask them your questions that you may still have. And I'll pass it on to Dr. Markman.
Yeah, just a very brief thought from an ethical perspective. We tend to be a little bit paternalistic when we select patients and say, well, we select patients that don't have another therapeutic alternative. They have difficulties with the assist device. They don't qualify for the allotransplant. But we could think, why don't we give patients a choice and say, even those who are on an assist device and are waiting for an allotransplant, why not give them the choice to stay on the assist device, wait for the allotransplant, or choose xenotransplantation. I think it's probably just too early now, but I think we should think about this as an option. Why don't we really, well, we need a good informed consent process, of course, if we enroll these patients, but why not give them a choice?
Great idea.
Yeah, I think those are very thought-provoking points, especially also in the context of the pediatric approach, because as Dr. Chinok nicely mentioned, a mother will always choose whatever we will offer them. So I think we have to have very high standards from an ethical perspective as well in thinking about those trials. Dr. Muhyiddin, did you want to have a last comment there as well?
Yeah, just I want to say that, you know, the one thing that was most important in this selection of this particular patient was, number one, that the main reason for his ineligibility for allotransplantation was his non-compliance. So I mean, you know, we can, I mean, that was, you know, was the main concern that our heart failure team used. The other thing is his own will to get the first ever big heart transplant. That was, you know, I mean, now we are receiving several emails of patients offering themselves for the next, you know, transplant. But to be the first one, I mean, that took tremendous courage from that patient. So I mean, you know, when we presented that option to him, and he said, you know, yes, I mean, you know, that, I mean, yes, there were some, you know, complications later on, which, I mean, that type A was never, you know, could have been suspected. But otherwise, you know, he was okay patient. I mean, you know, and he was young patient, although he was on ECMO. But otherwise, you know, if the type A dissection didn't happen and some other complications didn't happen, or, you know, later on infection doesn't happen. I mean, maybe I mean, he left for 60 days, but we should not ignore that, which is more than the first allograft lift, right? So I mean, you know, there is hope, and we think that this was a great success as far as we are concerned.
Okay, thank you so much to all of our panelists and speakers for this great session. I think it was very educational. Please, I hope you'll be open for getting questions from the audience. Thank you for listening in, everybody. Thank you for asking your questions. Sorry if we weren't able to address all of them. And this concludes our webinar for today. I don't think we have a final message from the CEO because we're already over time, but hope to see you around at the next meetings. Thank you.